Hello and welcome to the introductory video on fluorescence microscopy. But before I even start talking about fluorescence microscopy, we're going to talk a little bit about fluorescence. And in this short introduction, we're going to talk about what is fluorescence and what are some of the applications of fluorescence. So first of all, what is fluorescence? Well, fluorescence is defined as the emission of electromagnetic radiation, especially of visible light, stimulated in a substance by the absorption of instant radiation and persisting only as long as the stimulating radiation is continued. But what does this actually mean? Well, to demonstrate this, we are going to use something what's called a Jablonski diagram. So this here is called a Jablonski diagram, and as we all know, when electrons are at their neutral state, it's called a stage called ground state. So here the electrons are all nice and happy and there's no excess energy. When light or UV radiation or any other form of electromagnetic radiation is given to these, they will absorb this energy and the electrons will jump from a ground state to an excited energy level. And in here, the electrons have absorbed all of that energy jumping up. When they're at this excited energy state here, the electrons will be vibrating and giving off small amounts of energy. This could be as heat or micro amounts of radiation. All of a sudden, these can then jump back down to ground state, releasing energy equal from this energy state down to this, and this is often given off as light or other forms of radiation. However, when we decrease the energy from UV light down to violet light, as you can see that solution which was given off violet light is now back to colourless. This is because if any incident radiation is of equal to or less energy than what the emitted light would be, there would be no fluorescence, because the rule is you have to have greater energy than what could possibly be emitted. So another example would be to use indigo. So if I then shine an indigo light onto this, notice that the indigo solution has returned to colourless. So what do you think would happen if I shined blue light onto this? Well, as you would expect, the blue solution returns to colourless, but all of the other solutions, green and below, retain their colour. The same can be said with green. The green would go back to colourless. With yellow light, all we can see is the red and orange. With orange light, all we can see is the red. And if I shine red onto this, we get no fluorescence at all. Okay, so now let's talk about a few applications of fluorescence. So using modern technology, we can now use fluorescence in the diagnosis of certain diseases, but in particular diseases associated with E. coli. So here in this picture we have two microcentrifuge tubes, and one of them is fluorescing, and the one that's fluorescing actually contains trace samples of E. coli. This is because each of these samples have been treated with a chemical which, when in the presence of E. coli, begins to fluoresce, as we can see here. We can also use fluorescence in testing for contamination of water. So for example, if certain chemicals are in drinking water, we can then see if they're fluorescing, and if so, we can then figure out what chemicals these are. We use fluorescence in Western blotting for when we're using horseradish peroxidase, giving off a fluorescent byproduct. And as we will see in a later video about fluorescence microscopy. So just to summarise everything that we've been told, the incident wavelength must be shorter than the emitted. So this therefore means the incident wavelength must be of a higher energy or a higher frequency. The emitted energy is always lower than that which was received. This is because energy is lost by vibrations of the electrons. So this was the introductory video for fluorescence, and we found out what is fluorescence and a few applications of fluorescence. And we are now going to move on to fluorescence microscopy. Thank you for watching. This is called a dart box, which is used to block out any light which could interfere with any fluorescence which is being emitted from your sample. When inserting the slide, you remove the dart box, and as you can see here, the lenses are located. On screen up here, you can see the lens objectives, and here it tells you what lens you're using. The lenses go from 4, 10, 20, 40 and 60, with 40 being the one that is most commonly used. When placing your sample on, make sure you place the right side up, as you do not want any of your sample to contaminate the bottom of the microscope. You can then use these dials located here and here to help move the stage, to help align your image, and you can then use the ones on the side here to raise the platform to help focus the image. When you are ready, you can then place the dial box back onto the microscope, and then you can turn the light on to begin capturing an image. At the top of the screen, it tells you which light sources are being detected. For example, at the moment, DAPI is being detected. The one next to that is green fluorescence. The slides in this example have also been stained with DAPI and GFP, which stands for green fluorescence protein. Then, using the settings at the bottom of the screen, you can then adjust the focus and the brightness to help find and locate your image. Alternatively, you could also use the scroller on your mouse to turn the brightness up and down. Furthermore, you could also use the dials on the side of the stage to help move your image to help locate an appropriate spot. Once you're happy with the image you've got on the screen, you can then take a screenshot or capture of the image using the camera icon located at the bottom of the screen. If you have a memory stick plugged in, you can automatically save the image onto it. So in the example on the screen, it's only viewing what's emitting DAPI. So what you can do, you can change the settings to GFP to get an image of whatever is emitting green fluorescence. You can then therefore experiment with more of the settings to try and help focus your image. Once you're therefore happy with the image you've got on the screen, you can then reset capture to save the image you have got. Once the image has been saved, you can then toggle and overlay the images to give you a view of both of the DAPI and the GFP. From here you can then experiment with the contrast and settings until you've got a suitable image. You can then set a measuring scale for the size of the cells which you can then move around the screen. You can zoom further into the image by double-clicking the left mouse button, and you can zoom out by double-clicking the right mouse button. You can also open up a toolbar, and on this toolbar you can also set a time lapse of an image, so you can take a long exposure image of the cells. Furthermore, you can then also create a video of the cells interacting with one another. Another useful feature is the cell count, in which a grid is created and you can then click to mark and count the different cells you can see on the screen. Once you are happy with the image on the screen, you can then hit capture, and you can also change the save location. Furthermore, you can also change the format of the image to change to JPEG or any other image files. Another useful feature is the ability to increase the exposure time. By increasing the exposure time, you'll be exposing the cells to more light, and you can therefore build a more clearer image. As a result of this, the time taken for the image to load up will be considerably longer, as a result of greater clarity. Another feature is the ability to grayscale the image. So when you grayscale, you can also get these little red patches that appear in your cells. And these little red patches indicate overexposure. To get rid of these, you can then just change the brightness lower, which can then correct this issue.